Right, welcome back for the evolution of cephalopods. And as always, we're going to lead off with that little plectrunoceros. Okay, so this is from a nice paper looking at some theories on the origin. I'm mispronouncing his name. It's Jerzy Zeke, and I'm mispronouncing both words of that. But very good paleontologist, works on not only mollusks but also some of the earliest organisms from the Ediacaran. Done some very famous work on there. And what he's pointing out here is that these organisms all share a common starting point. So these monoplacophorans, he's saying, are probably most closely related to the earliest branches of the mollusks, at least the ones we have records for that had a shell on the outside. No telling how many mollusks there were before shell biomineralization really got going, because soft tissue preservation is not great before that point. Anyway, you can see the little bulge down here. It's like a little cup. That seems to be a common feature. You'll find that in the early bivalves. You'll find that in the things that lead up to cephalopods. You will find that in the early cephalopods and bactritoids and all that sort of stuff. You'll find that in bellerophontids, which are like a sort of swimming gastropod, we think, or somewhere in between gastropods and something else. It's hard to be sure. And it would be the innermost little chamber of gastropods. Scaphopods are the tusk shells, and we're not really sure how those evolved, but there is a, I think, reasonably convincing line that they come from bivalves, which lead to a thing called rostroconchs, which would then lead to scaphopods. I think visually it makes sense. I have no idea if, in terms of the muscle structure on the inside and the placement of the internal organs, if that makes as much sense as I hope it does. Yeah. All right. So we mentioned before that snails commonly have an operculum, and here's some nice little, let's see, C and D are operculums from gastropods. And so it looks like it's sort of a flat, snaily shell, and that's just because they're growing it. Every time the animal grows, it needs to make a larger door for its house, because its house just got larger. Yeah, it makes sense. And the door has to cover the entire opening. Yeah, that also makes sense. On nautiloids and quite a range of cephalopods, you actually get a modification of this. So we think it's homologous, like from a common ancestor with the operculum of snails. But these end up being used as a sort of a three-part jaw. And so here you can see a nautiloid. There would be the eyes on the side, and the two opercula are down here underneath this muscle, here and here, and then the third one is going to act like a beak, so that these things can tear into flesh and just murder stuff or rip apart dead stuff. In the case of Nautilus, then and now, it would seem. All right. So back to ammonites. And ammonites, you remember, the big difference is they're sifungals on the outside, whereas a nautilus, it runs right down the middle. Do ammonites have this organ? The organ is called an aptychus. There we go. Let's make that bold. Nice. Looking good. And let's make it colorful. Yeah, that's nice. Okay. Yeah, you can see it here in place on the organism. So this one has pulled its aptychus shut like a little door, because why shouldn't your jaw function as a door if you need it to? Yeah. Here you can see one that's laying out, so the third jaw part is underneath, and I think they photographed it with UV light or something to get that to come out. And here are the other two parts that would be closing, and so that's what you're looking at here. The other jaw would be underneath right there. This is a weird fossil. This is currently, I think, the only really clear evidence we have for the internal organization of an ammonite, but I would expect there will be more at some point. Yeah, just bound to happen. So what we've got here is all the soft parts and the aptychus, like the jaw of the ammonite, but none of the shell. And so the question raised in the title of this paper is this like failed predation, like something came along and crunched the shell, and then the the soft parts, dropped out of the shell and basically died, probably from compression. Um, or is it a case of weird, like a thing that happens after death, like they build up enough gas inside the the shell with this rotting corpse of an ammonite that just sort of fires like a cannon and pops the guts out that fall in a little pile outside. Either way, not pleasant. Not pleasant for the ammonite, not pleasant for you or for me. Yeah, too bad. But we've got the entire organism here, and we've got enough of the organism they can actually find a male parts on here, a vas deferens and a penis, and so you can tell this was a male ammonite, and they were sexually dimorphic. Yeah. And so, fitting all those parts back in, they photographed it with different types of light, which will pick out differences in some of the minerals, which you will often get on different organs. It's amazing, and it is very fortunate that that happens. So, here they've reconstructed it as they are laid out on the ground there. And then they've kind of put it into the shell from the same species that they find at that spot. So they're showing where the nice eyes would be, little eye capsules, and where the jaw would fit, and where these organs would be. And so look, this would be the back end of where the phragmocone is. This would be the front end. And so you see the phragmocone is more than half a turn around the outside of this particular size of ammonite. And others may have been a little different, but that's probably fairly close to representative. So the actual male parts, the reproductive organs, so testes are up here. The penis would be right there, and it's actually going out into this canal. So those are the gills. That's basically the breathing canal, but the siphon is pulling in water here and spitting out water from here. And so they're essentially releasing their gametes into their lungs, and they are then squirted out into the world. So good job. All right. So here again from the Zeke paper, the evolutionary relationships between some of these groups. So there is still an open question. It is definitely not resolved. This is one hypothesis, and it's a decent hypothesis, but there are other hypotheses out there, and frankly, we need more fossils from early cephalopod ancestors to know for sure. The idea here is that there are, and these do exist, halcyonellids that are really tall, like this. And they would have little chambers on the inside. The chambers don't have a siphuncle, and so we would call these halcyonellids. If you look at these, though, you can actually find little dents in the shell that tell you the organization of the muscles in there. And those are organized the same way as in a modern monoplacophoran. So they're probably related to those little things, the one shell primitive guys. And the idea is all you got to do is put some kind of little hole in this chamber. But, you can wish yourself to evolve a hole in your bones, and good luck. It would take some kind of genetic accident or some kind of unusual genetic event, maybe, for this to happen. And it's kind of a miracle that the organism didn't die. But once you have partly closed off chambers, as long as there's a little bit of that flesh that can eventually evolve into a siphuncle that's running through those, then there's the possibility of depth control and of becoming a predator that will drop down sort of in the water column and pounce on something. And that seems to be what they were up to. Here's the thing with the muscle patterns. And so these are your monoplacophorans down here in the group Triblidida. These are a group that are on the way to becoming nautiloids, but are a little bit funky. Hyolithids are a group that people have tried to stick on the mollusk tree. I think they probably do belong on a mollusk tree. They've also tried to stick them as an early type of brachiopod, but that has now been pretty widely discounted. The field generally doesn't believe that anymore. Although you can find some papers if you look back, and if you ask ChatGPT, it may still tell you. Depends what it's read. But the nautiloids up here, some of those will actually have the same sort of pattern of these muscles, little scars running around in a circle. And so the idea is that, yeah, they are showing their original relationships through this. Okay, so another hypothesis presented in that same paper is that you take something that is like a halcyanellid or like a yokel cyanellid. Here's yokel cyanella, and this is the one that's like, it kind of looks like a snail. It's a cap-shaped mollusk, but it's got a tube on the back, which may be for a siphon, maybe for exhalant current, maybe on the way to becoming something like a scaphalopod, because they would have a similar organization to this, although very sort of skinny version of that. Then the idea is that, yeah, if you take something like this, it could get taller, and it's this little spout coming out here. If you just learn how to remake that shell on the inside as a way to grow, then that little chamber, that little spout, becomes the siphuncle tube. Possible. The other idea is that you make walled off chambers first, like in Nitoconus, and just develop, or in a Cercothecid, and you just develop a hole in those. That's a tough call. Can you just develop a siphuncle like that? I don't know, because it would make more sense if it was already there and doing something else, and it's just being repurposed. So I like this path over on this side more than I like these two paths, but doesn't matter what I like. It matters what's real, and that hasn't been demonstrated yet conclusively. Okay, I think this one's the last one in this section. This is just looking at the siphuncle placement in the early development stages of different groups of cephalopods. And so here you can see it in the nautilus, looking great. And you can see this is also the nautilus over here, running through the middle of the chambers, just like it does. And here you can see some of the other groups and these very convoluted sorts of siphuncle structures that you get in some of the earlier cephalopods. So which of these becomes, most successful? Do multiple of these go on and found other groups? There are people who may be willing to argue for either of those. But yeah, I just thought it'd be nice to show you what's going on here. And so from the siphuncle placement, and particularly from the embryonic, like the early development siphuncle placement, that is the best surefire way to diagnose what kind of cephalopod you're looking at. And to do that, you unfortunately have to section the thing, basically cut it in half and polish it off. And then you can see just what it was doing inside. So yeah, somewhat destructive, but very revealing. All right, come back for the next section, where we talk as a shorter one about some of these features on cephalopods and what they do, and then that's going to be a wrap for cephalopods.